हॅलो सुरू करा.
हो सर. A very good afternoon, ladies and gentlemen. President of this 1 month classroom program, Dr. Punjab Chavan, teachers who will deliver this 1 month countrywide program, coordinator of the different subject for this countrywide teaching program, and the student participant. Dear friend, I warmly welcome you all in this 1 month countrywide classroom teaching program. At the outset, I welcome you all in this program. This event is organized by Faculty of Science and ACS of Digambarrao Bindu Arts, Commerce and Science College, Bhokar. Dear friend, we are very young as a science faculty, as our institution has initiated GSC courses. BSc courses okay. Okay. Dear friend, I'm very sorry for the disturbance. We are very young as a science faculty, as our institution has initiated BSc courses in 2010 and 11, and since then we are working tirelessly to guidance under the guidance of the president of our institution. Scientific point of view and the student of dear friend, this year we are afraid about to continue our daily routine learning process due to the pandemic the COVID-19, and we have different guidelines from the government to continue our teaching learning process or to this COVID-19 lockdown. In response to the situation, have been instrumented finding new network is unstable. Sir, network unstable hai. Okay, sir. Thank you. So, in response to current situation, educators have been instrumented in finding a new way to ensure learning continues for our students by developing online and offline learning materials, learning about the working of the video conferencing tools to be able to meet students regularly. In concern with the above situations and above things, our accuracy department and the Faculty of Science has initiated to run this countrywide classroom program to meet the requirement of students in this difficult situation. From the concept of the University Grant Commission, UGC New Delhi, countrywide classroom program, which was launched in 1984. Higher education. UGC began its transmission of countrywide classroom program from the August 1984 through the our television called as Doordarshan, the national network. Initially, the coordination with this center was done from the UGC office with the support of a consultant, and subsequently, as an inter-university center named as the Consortium for the Education Communication, that is a CEC, was set up in the year 1993 to coordinate with the media center and to make this countrywide classroom program mission most effective and successful. Similarly, we are trying to run this countrywide classroom program 2020 on a virtual platform like Zoom to meet the requirement of our students. But the main vision of this countrywide classroom is to electronically reach a large number of students, teachers and general public with the quality education material, so as to address the issue of the accessibility of a higher education with quality and quantity through visual and through virtual platform and through the YouTube channel. The main aim of this countrywide classroom program is to spread knowledge and expertise of the academicians working in the various departments of our university, colleges, and different institutions of higher education to the virtual platform of the students. For this countrywide classroom program, we have initially asked our faculty member to participate, and from them we have received huge response for this countrywide classroom program. On the basis of UPL response, we are organizing this 1 month countrywide classroom program for the student of our SRT and all the universities in the Maharashtra, as around 70% of the syllabus from all university in India are similar. For this countrywide classroom program, we have received responses from all over the India. Total till date, we have 48 faculty teachers are enrolled for this event, and in meanwhile timing, many of the teacher faculty will be enrolled as per the time slot allotted to them. Dear friend, without taking your much time, I conclude my introductory talk about this CWCP program, that is countrywide teaching program, and I call upon our beloved principal sir to address as a presidential address and inaugurator for this countrywide classroom program, Dr. Punjabrao Chauhan sir, please.
नमस्कार
Sir.
आवाज येतोय ना? हॅलो हॅलो
हा येतोय का?
हा
सर येतोय.
हा नमस्कार. कंट्रीवाईड क्लासरूमच्या आजच्या या सुरुवातीच्या प्रोग्राम मध्ये ही लेक्चर सिरीज जी महाविद्यालयाच्या वतीनं आयक्यूसीच्या डिपार्टमेंटच्या वतीनं घेतली जाणार आहे, त्या लेक्चर सिरीजचं उद्घाटन पहिल्यांदा झालं असं मी जाहीर करतो. या कार्यक्रमाच्या निमित्तानं वेगवेगळ्या महाविद्यालयातून महाराष्ट्रातल्या अनेक तज्ञ प्राध्यापक आपल्याला मार्गदर्शन करणार आहेत, वेगवेगळ्या विषयाच्या संदर्भामध्ये मार्गदर्शन करणार आहेत. आपण सर्वजण त्या मार्गदर्शनाचा योग्य असा लाभ घ्यावा, जेणेकरून आपल्या ज्ञानामध्ये, आपल्या विषयाच्या ज्ञानामध्ये चांगल्या पद्धतीने भर पडू शकेल. रोजच्या टीचिंगच्या माध्यमातून आपले दैनंदिन क्लास आणि आपल्या महाविद्यालयातील टीचर आपल्याला शिकवतच असतात, परंतु इतर टीचरच्या माध्यमातून जे शिकवलं जाणार आहे त्यांचं ज्ञान आपल्यापर्यंत पोहोचवलं जावं, त्यांच्या तज्ञ टीचिंगचा अनुभव आपल्यालाही घेता यावा, या दृष्टिकोनातून ही सिरीज या ठिकाणी आयोजित केलेली आहे. विशेषतः ही सिरीज अनेक दिवस चालणार आहे, त्यामुळं विद्यार्थ्यांनी या सिरीज मध्ये सहभागी होणाऱ्या सर्व प्राध्यापकांचे तास जे आहे ते तास न चुकवता अटेंड करावे, जेणेकरून त्याचा लाभ आपल्या विषयाच्या ज्ञानामध्ये भर पडण्यामध्ये होईल आणि त्यामुळं आपलं ज्ञान सर्वांगीण विकसित करण्याच्या दृष्टिकोनातून त्याचा फायदा होईल. आयक्यूसी डिपार्टमेंटनं अतिशय नाविन्यपूर्ण प्रयोग विशेषतः आपल्या विद्यापीठातच नाही तर मराठवाड्याच्या लेवलवर असेल असा प्रोग्राम कोणत्याही महाविद्यालयानं आयोजित केला नाही, परंतु तो आपल्या आयक्यूसी डिपार्टमेंटनं दिगंबरराव बिंदू महाविद्यालयानं आयोजित केला. त्याबद्दल आयक्यूसी डिपार्टमेंटचे प्रमुख अरविंद चव्हाण, सायन्स विभागाचे प्रमुख डॉक्टर सचिन तावडे आणि सर्व सायन्स विभागातील प्राध्यापकांचं मनपूर्वक मी अभिनंदन करतो. आणि या सर्व कार्यक्रमाच्या निमित्तानं, या सिरीजच्या निमित्तानं आपली ओळख, आपल्या महाविद्यालयाची ओळख महाराष्ट्रभर होईल. आणि यातून जे काही लेक्चर, चांगले चांगले लेक्चर या ठिकाणी होतील, त्या सर्व लेक्चरचं आपल्या YouTube चॅनलच्या माध्यमातून डाउनलोड करून त्यामध्ये लोकांना पुन्हा एकदा पाहण्याची, विद्यार्थ्यांना पुन्हा एकदा ऐकण्याची संधी त्या ठिकाणी प्राप्त करून देता येऊ शकेल. हा अतिशय नाविन्यपूर्ण प्रयोग आपल्या विद्यापीठाच्या माध्यमातून महाविद्यालयानं सुरू केला. त्याबद्दल मी महाविद्यालयाच्या या सर्व सहकार्याचे मनपूर्वक अभिनंदन करतो आणि पुन्हा एकदा या सिरीजचं उद्घाटन झालं असं जाहीर करून माझे 2 शब्द संपवतो. जय हिंद.
धन्यवाद. या कंट्री वाईड क्लासरूम प्रोग्राम मध्ये सर्व विद्यार्थी या कंट्री वाईड क्लासरूम प्रोग्रामचा लाभ घेतील आणि आपल्या विद्यापीठात जो कॅम्पस आहे तो आपण या कंट्री वाईड क्लासरूमच्या अनुषंगाने त्या विषयाचा सिलेबस हा पूर्ण करण्याचा प्रयत्न पण आपण इथे करणार आहे. आणि विद्यार्थ्यांना एक चांगली संधी इथे आहे की आपण एका महाविद्यालयामध्ये असताना, रेग्युलर क्लासेस असताना आपण एकाच टीचरचं मार्गदर्शन आपल्याला त्या विषया संदर्भाने जे काही टीचर आहेत त्यांचंच मार्गदर्शन आपल्याला मिळत असते. पण या कंट्री वाईड क्लासरूमच्या निमित्ताने आपल्याला वेगवेगळ्या तज्ञ शिक्षकांचं महाराष्ट्रातीलच नाही तर पूर्ण भारतातील जे काही टीचर आहेत त्या विषयाचे, तर त्यांचं मार्गदर्शन सुद्धा विद्यार्थ्यांना इथे लाभणार आहे. वेळ न घालवता मी इथे आजचे प्रथम सत्राचे मार्गदर्शक श्री सचिन भोसले सर यांना विनंती करतो की त्यांनी आपलं प्रथम सत्र जो काही आपला टॉपिक आहे एपिकल्चर हे सुरू करावं. सचिन भोसले सर.
Good afternoon, sir.
Sir, very good afternoon.
Thank you, Dr. Arvind Chavan, sir, and all the organizers
of
this program for giving me this opportunity for talking to you people. I'm going to share my screen.
Yes, sir. Please. Now you can share.
Can you all see my screen? Am I clear? Hello, sir.
Was there any issue?
Is my screen visible to you all people? No? Sorry for the disturbance.
Is there any problem?
Is my screen visible to you?
No, sir.
No. Okay. I'll get it back.
Sir, can you share us the PPT? So we have to share it from outside.
Okay, okay, sir. Okay, sir. That's what I do.
Just
I will
WhatsApp.
share it to you. I will share it to you.
Yes. Oh, okay, okay. Sir. Next.
This must be the because of network issue. Sir, I have shared with you. Chauhan, sir. Chauhan, sir. I have showed my PPT with you.
Sir, you are sharing with me or you are sharing yourself?
I have shared with you. I'm not
Oh, okay, sir. Just sit. On WhatsApp?
on WhatsApp.
Yes, 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 yes.
I'm not being able to share my screen.
Mm-hmm. Yes, sir. It is not visible.
Why it's not visible?
Okay.
Okay.
Only white screen is visible. Yes, yes, sir. Just
Okay,
try one
I'll share
second.
it.
And just open your PPT on the desktop, sir. See, sir.
Yes, sir.
First,
Yeah.
you open the PPT on your desktop, or allow me access to your desktop.
So I'm presenting this from my mobile actually. I'm not in the lab. Yes, sir. I have shared it with you. PPT.
Okay, okay. No, and right now you can share, sir.
Okay, okay.
Sir, try once again.
Okay, I'll do it, sir.
Yes, sir. Try once again.
Sorry for the disturbance. Okay. Sir, can you share my PPT?
Yes, sir. I am sharing your PPT. Just
Okay, sir.
wait a moment. Okay. Yeah, your picture is visible, sir.
Okay, just a minute. I'm getting. Okay, share my PPT, sir.
Just I'm downloading it.
Hello, sir. Are you doing it, sir?
Yes, sir. I am downloading it.
Yeah, network issue. I also have the
Just
network
wait a-
same issue.
Yes. Oh, okay, sir. I'm trying from my side.
Okay. Is any problem, sir?
That is opening, sir. You have shared. Okay. Now it is visible, sir.
Yeah, it's getting.
Okay, sir. Just go ahead, sir.
Okay. Okay. Good afternoon, everybody. Myself, Sachin Bhosle, from Abhinav College of Science, Akolai. I'm presenting to you the topic, which is entitled as Introduction to Apiculture. Apiculture. Next slide, sir. Let us know about the economically important social insect, that is honeybee. We all know about what is honey. You know honey? that's a versatile food material that we all use in our day-to-day life. Mostly it has many medicinal properties, like in case of vomiting, in the case of cough, we mostly use the honey. It is sweet in taste. Where this honey comes from? Where does this honey come from? Do you know? Yeah, you guessed is right, that it comes from the honeybees. Honeybees. Honeybees are the social insect, which is highly organized social insect which live in colonies. What name mostly? What's the apiculture? That we have seen about the honeybee. And what's apiculture? Previous slide, sir. Apiculture. Apiculture is nothing but the rearing and management of honeybee. Yeah, sir. This side. Rearing and management of honeybees for the production of honey, bee wax, royal jelly, bee pollen, and propolis. Not only for these commercial products, but honeybees are rare. Mostly nowadays they are rare, or mostly for the pollination purpose. Pollination, in case of plant pollination, to increase the crop yield. That mostly use honeybees. And apiculture is nothing but the rearing and management of the honeybees for the production of bee wax, honey, and pollination purpose. This is the apiculture. A beekeeper, it's called the person who keeps bees and manage them. The person is called as apiarist. The person is named as apiarist. And location. Location means the place where the bees are kept for their management. The location, the place is known as apiary. The scientific study of bees, honeybees, this branch of science is known as apiology. Next slide. Next. That you got. What's apiculture? That is scientific, nothing but the scientific study of honeybees for their rearing and management. Let's look at the systematic position, that is classification of the honeybees. Okay, it belongs to kingdom Animalia. What is the general features of the kingdom Animalia? That is, chlorophyll pigments are absent in the kingdom Animalia. Then phylum, it belongs to phylum Arthropoda. Arthropoda, it's a class. Arthropoda, phylum Arthropoda. What is the basic general characters of phylum Arthropoda? They're having jointed appendages. Then it belongs to class Insecta. What is the peculiar feature of the class Insecta? Class Insecta are also known as hexapods. Hexapods, they're having 3 pairs of legs or 6 legs. And body is divided into 3 regions, that is head, thorax, and abdomen, and belongs to order Hymenoptera. General character of Hymenoptera, that is 4 membranous wings are present in them. The organisms which belongs to order Hymenoptera, having 4 membranous wings, not pairs, 4 membranous wings are present. Then family Apidae, genus Apis, and there are 4 main species of honeybees which are used in the apiculture. 4 main species, which are they? The species are Apis dorsata, Apis indica, Apis mellifera, and Apis flori. Apis dorsata, which we know that it's giant bee or rock bee, which is not used in case of apiculture. It is mostly used in rocky areas. That's why it's called as rock bee. Then Apis indica, it is indigenous bee. That means it belongs to, it's originated in our country, that is in India. Then Apis florii. Apis florii is a little bee. Apis florii, which is not used in apiculture. Next species, that is Apis mellifera. It's mainly exotic species. That means it is not originated in India, but it is Italian, it is also called as Italian bee or European bee. Next. Let's look at the history of apiculture. Apiculture, that is collection of honey. Let's look at the history, that is collection of honey from the honeybees was done before 13,000 before 30,000 years before Christ. Medicinal importance of the honey, we know that is from prehistoric time. It is also described in our Vedas, Puranas, and Ramayana and Mahabharata. It has been having medicinal properties. That's why it is also used in Ayurvedic medicine. It having medicinal properties. It is described in various Vedas and Puranas. Next slide, sir. Next slide, sir. Okay.
Yes, sir.
Species of honeybee. The previous slide. First species, that is Apis dorsata. Apis dorsata is known as rock bee. This is the largest species of the honeybee that we know. We have the family of four species that we have seen, that is Apis dorsata, Apis indica, Apis florii, and Apis mellifera. Apis dorsata, that is a rock bee. It is the largest honeybee, which builds single vertical and high branches, single vertebral comb on the high branches of the trees and rocks. It builds combs on the rocks. That's why it's called as rock bee. It produces a large quantity of honey, but these bees, rock bees, they are very gregarious. So they are very difficult to domesticate. They are not domesticated at any cost because they are very gregarious. Then next, that is our indigenous species, Apis cerana indica. Their common name, that is Apis, the Indian bee. Then it is a medium-sized bee. It is mostly used in apiculture. Apiculture, that is beekeeping. It is mostly used in apiculture. Then next, hive, that is comb of the Apis indica, is composed of several parallel combs on the branches of tree. The bee produces large quantity of honey, which is easily domesticated. This is the mainly used bee species, that is Apis indica, which is used in apiculture or beekeeping. Next slide. That is next. That is Apis florii. Its florii is known as garden bee, small pollinating bee, or little bee. It is not getting domesticated or not used in the apiculture because honey yield of this species, Apis florii, honey yield is about 1.5 kg per colony per year. Honey yield is very poor. That is half kg per colony per year. That's why it is not used in the apiculture or beekeeping. Next species, that is Apis mellifera. It is known as European bee or Italian bee. It yields honey about 8 to 10 kg per year. They are small-sized bees. It builds single small combs in the bushes. 3 bushes, it builds a small combs. It is this species of Apis mellifera, mostly used in apiculture. In India also, this Apis mellifera are used for the production of honey. It yields honey about 8 to 10 kg per colony per year. Next species. Next slide. Let's look at the life cycle of the honeybees. In case of life cycle, honeybees, we know that honeybees is a colonized or highly organized insect. In case of colonies, it lives in colonies and having 3 different caste systems. That is 3 types of, that is queen, drone, and workers. Queen, drone, and worker. Its honeybees shows complete metamorphosis. They are termed as holometabolous insect. What is meant by complete metamorphosis? That it may show 4 different life cycle stages, that is egg, larva, pupa, and adult. The insect which shows all these 4 stages of life cycle, they are known as holometabolous insect. Let's know about the queen bees. Queen is the only fertile female of the colony. Only fertile female of the colony. She is the largest in size as compared to worker bees and drone bees. There is absence of wax glands. Wax glands are absent. We know that it's wax which is used by honeybee for the making comb or hive preparation or hive construction or comb making. Wax is used. Lifespan of the queen bee is about 3 to 4 years. It lives for 3 to 4 years. The only function of the queen in the colony is to laying eggs. That's why it's called a queen. And only function of the queen is to lay the eggs. It lays about 1,500 to 2,000 years per day in the egg-laying period. Next. Drones. Drones are called as king of the colony. Drones are called as king of the colony. Their lifespan is about 60 days. Wax glands are absent in the drones and sting also absent in the drones. Sting apparatus, which is protective apparatus or protective weapon of the honeybees. Drones are developed from unfertilized eggs. Queen lays 2 types of eggs, that is fertilized eggs and unfertilized eggs. Queen and the workers are produced from the fertilized eggs, and the drones are produced from unfertilized eggs. Worker bees, the main feature, all the duties on the honeybee colony belongs to workers. They have more type of duties than the queen and the drone. That queen, the main function of the queen, that is laying eggs, and main function of the drone to fertilize the queen. The only function that performed by both, that is queen and the drone, that is performed by drone and the worker, that is sense of temperature. In case of dry and cold conditions, To regulate the temperature, they both, that is, drones and workers, through the beating their wings, movement of their wings, they maintain the temperature in the honeycomb, their hive. This is the only common function which is performed by drones and the workers. Then worker bees. There are about 2,000, 20,000 to 30,000 worker bees are present in a single colony of the honeybees. They have strong wings, strong membranous wings, and lifespan about 35 days. It lives for 35 days. Wax glands and sting apparatus is present in the worker bees. That we have seen, that is, it is absent in the drones, and which is present in the queen bees and worker bees. Then worker bees are developed from fertilized eggs. Fertilized eggs produce queens and the workers. And they have well-developed sting, and hind legs are with pollen sacs. Pollen basket is present at the hind leg. The main feature of the hind leg, that's due to presence of pollen basket, feature that is collecting pollen from the various flowers. Pollen baskets are used for the storage of pollen, which is collected by honeybees from the different flowers. They perform different duties. Worker bees perform different duties as they are followed. For first, in their life cycle, for first 1 to 14 days, that activity of the workers bees, that indoor activities. They perform the indoor activities. Indoor activities, in that case, which are the indoor activities, that is cleaning the hive and feeding the larva. For first 1 to 14 days. Then 14 to 20 days, worker bees guard the hive from the entrance of the enemies. Guard the hive. Enemies. Which are the enemies? That is wax moths. Wax moths are king crows and all the birds that guard the hive from the predators. On the day 21 to 35, they perform outdoor activities, that is foraging. In case of foraging, they collect nectar and pollen for the preparation of honey. For the preparation of honey, they collect nectar and pollen from the flowers, from the surrounding area. Next, we have to see different products from the agriculture. There are the first main product that we all know, that is honey. How does bees produce honey? That is, bees produce honey from the nectar of the flower. The flowering plants have the nectar, which is collected by the honeybee, and through the enzymatic activity and water evaporation to the regurgitation of the pollen, the honey is produced. Then medicinal properties or uses of honey. Where honey is used. Honey is used since prehistoric time. It has medicinal properties and it is a versatile food material which is highly nutritious, which is used by human beings. That is, it's rich in energy, minerals, and vitamins. That is vitamins B, vitamin C, medicine, In medicine, it is used to prevent cold, cough, and fever, which we largely use honey in case of our fever, cold, and cough. When we have cough, we mostly use honey to prevent cough. That is, let's see the chemical composition of the honey. Honey contains sugar, various different types of sugar, that is levulose. Levulose is known as L-fructose sugar, which is about 38.9, it near about 40%. 40% about is levulose, that is L-fructose sugar. Then dextrose, dextrose sugar, also known as dextrine, which is about 21% in the honey. Then various different types of sugars, that's maltose and other sugars like glucose, which is about 8.81%. Then enzymes and pigments are about 2 to 2.21%. Then water is about 17% in honey. This is the chemical composition of the honey. Next. Next, bee product, that is bee wax. Bee wax is secreted by wax glands of the worker bees, which is located at the last 4 abdominal segments, that is 4th to 7th abdominal segment, where the wax glands are present in the worker bees. Uses of bee wax. Uses bee wax used in making candles, various cosmetics, ointments, and polishes, that is mostly used by polishes in case of nail polishes. It is used in nail polishes, cosmetic, ointments. It's having medicinal properties. It's also used in lipstick, preparation of lipstick. In honey bee wax is used by honeybees for the preparation of, for the formation of comb foundation, for the construction of comb. Bee wax used by the honey. Next slide. The most important product, that is royal jelly. It's a royal secretion. For first 3 days, that is larva of queen, drone, and workers. For first 3 days, they all are fed on the royal jelly. And from 4th day, only the queen, the larva of queen or queen is fed throughout the life on the royal jelly or royal substance. And other larvae are fed on the bee bread, which is a mixture of honey and pollen. Mixture of pollen, nectar, and honey. It's called as bee bread, which is produced by royal jelly. It's a glandular secretion of young worker bees. Worker bees of about 4 to 10 days old, produced by hypopharyngeal glands of the head. Hypopharyngeal glands of the head of worker bees produce royal jelly. Uses of the royal jelly. It is used in royal jelly. It is used in treatment of diabetes, osteoporosis. Osteoporosis means it's concerned with bones. For the bone repairment, it is used. Mostly the film stars and other people also mostly use royal jelly for the skin glow, for the glow of their skin, because it having medicinal properties, and it is very effective for the glowing of the skin. It is also used in healing wounds. And it boosts immunity. It acts as an immunity booster. Next bee product, that is bee venom. Bee venom, we know that is honeybees, the worker bees and queen bees of the honeybees, that is having sting apparatus. Sting apparatus is nothing but the extension of the ovipositor. It is modified ovipositor, also known as sting apparatus. When we got the bite from the honeybee, we must observe the sting over our skin. When she bites, that is worker and queen bee bites us, it dies, because due to the biting, the ovipositor of the worker comes out along with the sting. Due to the bite, that's why due to the bite it gets died. Bee venom, it is bitter colorless liquid. It is colorless liquid. It contains various proteins which causes local inflammation. We all must got bite from the sting of the worker bee. It gets eventually died, but it contains venom that causes local inflammation of our skin. When bee venom is used in arthritis, for the treatment of arthritis, then nerve pains, then sclerosis, for the treatment of sclerosis, and desensitize the people the allergic to the bee sting. Some peoples have the allergies of the bee sting or bee venom. Due to the bee venom, there are some cases which, due to the allergic reaction, due to the multiple stings of the multiple bees, some people also get died due to the allergic reaction of the bee venom. Let's see the composition of the bee venom. Bee venom contains melatonin, that is about 30 to 50%, then phospholipase A about 10 to 20%, then apamine, it is about 3%, then hydroxidase, it is 2%, and various acids. It contains mainly proteins and various acids. Next bee product, that is bee propolis. What is bee propolis? Bee propolis is the resinous mixture of honeybees produced by mixing of saliva and bee wax with the exudate gathered from the seed buds. Bee propolis is the resinous mixture of the honeybee. Bee propolis. Let's see the bee propolis uses of the bee propolis. Bee propolis is used to seal the unwanted spaces in the hive. It's used to fill the cracks and crevices of the honeycomb. And it's also used to treat cough and throat irritation of the human being, it having medicinal properties. That we have seen. Then there are various diseases and pests, predators which attacks the honeycomb. Let's see the two types of diseases it's having, that is nosema disease and Akari disease. Nosema disease is caused by Nosema apis. Nosema apis. It is a protozoan which is caused Nosema disease to the larva of the honeybees. The main effect of this Nosema protozoan that is observed, there is black spots on the body of infected larva. The larva which has got infected due to the Nosema protozoan having black spots on the body. The next disease, that is Akari disease. It is caused by mite and having shrinkage of the larval body. Next. Next. Okay, that's thank you. Thank you, Dr. Alvin Chavan, sir, for giving me time to talk with you beautiful people and listening my presentation carefully. Thank you, sir.
Thank you very much, Sachin Bhosle, sir. Thank you for a nice presentation on apiculture and its life cycle. I think that we have apiculture as a paper in skill enhancement code in SRTM University. It definitely help our ACS student. And now, once again, I thank you for your nice presentation and hope to see you again in this countrywide classroom program with another lecture. Now, I call my colleague Dr. Jayawardhan Balkande, sir, to propose vote of thanks for this first session. Dr. Jayawardhan Balkande, sir.
Thank you, sir. May I audible, sir?
Balkande, sir.
Hi, yes, sir. May I audible? Hello.
Yes, sir. Yes, sir.
Yes.
Yes, sir. You are audible.
Yes, yes. Thank you, sir. Thank you.
Yes, sir.
Thank you, Dr. Sachin Bhosle, sir. It was a very nice and informative session, and you are elaborate all these aspects in apiculture, from its merit to demerits, the economic importance, and the venom and their so number of uses you have elaborated in your lecture. I hope this lecture is really helpful to all the students who have joined in our Zoom platform as well as in YouTube live also. So on behalf of Internal Quality Assurance Cell and Faculty of Science, Digambarrao Bindu Arts, Commerce and Science College, I am thankful to Dr. Sachin Bhosle, sir, for giving a wonderful lecture on apiculture. I hope he will also join with in our future events. With this, I again thankful to the organizer by organizing such a wonderful session, wonderful lecture of the Sachin Bhosle, sir, in the field of entomology, that is apiculture. In our university, the apiculture is one of the paper for the skill enhancement course, and he's really giving a wonderful knowledge to the all students. Once again, I thank you to the Dr. Bhosle, sir. Over to you, Arvind, sir.
Thank you, sir. Thank you very much. Dear friend, now we are moving towards another session number 2 for today's and the last session. And I welcome Dr. Rajan Wale, sir, who is associate professor in microbiology, and his topic for presentation is immunology. That is the major histocompatibility complexes first. That is topic especially for immunology and for the microbiology students. I welcome Dr. Rajan Wale, sir. Dr. Wale, sir.
Am I audible, sir?
Yes, sir. Yes, sir. You're audible.
Thank you very much, sir. Thank you.
You're most welcome, sir.
Should I start, sir? Chavan, sir.
Yes,
Hello?
sir. Go ahead, sir.
Yes, sir. Yes, sir. You have to start your session, sir.
Okay. Oh, thank you so much, Dr. Chavan, for giving me this opportunity to deliver a talk on the very interesting subject of immunology, and that is major histocompatibility complex. I have divided this particular unit into two parts, and that is why part 1 today and part 2 will be later. So immunology in its own way is very interesting, and that too MHC is a very important aspect of immunology. So in these two parts I will try to cover up the topic that is allocated in class of third year BA. So I will start my presentation. I will share my presentation, sir. Just give me a
Yes, sir. Sure, sir. Take your own time, sir.
Is my screen visible to you, sir? Someone, sir?
No, sir.
No, not it.
No, sir. Right, not now. But it is looking. It is looking that you are sharing your screen.
Okay, it may take a while. Wait.
Yes, yes. It is visible, sir. Yes.
It is visible? Okay.
Yes,
Is
sir.
it?
Yes, sir. It is visible.
Just.
Yes, sir. Yes, sir.
Okay. Hope I've got audience which is interested in immunology, the very versatile subject of life sciences, and it's in my as we deal with so many when we talk about uh, antigens and antibody reaction, even PG students friends, they think it as a diagnostics or only as a method of diagnostics, but immunology is a multifaceted subject. It has applicabilities in so many other relevant and parallel branches of life sciences as well. Sir, I am audible? Chavan, sir?
Yes, sir. Yes, sir. You are audible. Yes, sir. You are audible, sir.
Some message. Okay, some message appeared on my screen. So please let me know if I'm not audible. So
Oh, okay, sir.
hello. So
Yes, sir.
immunology is a yes, sir. So it has been said by some microbiologists that antigen antibody reaction is the most specific biological specific reaction on the earth. So this specificity we microbiologists and biotechnologists have exploited for several purposes, right from a diagnosis of certain diseases, treating certain infectious diseases, treating some deviated physiological conditions, or what we call as autoimmune or wrong immune responses, and so on. Nowadays, because of this COVID situation, at even in public places, at corners, nooks and corners, where we heard the terms like herd immunity, vaccination, antibody, and so on. So it is very the context of our subject is very much related today, even for the situation that we all are facing. So immunologists and microbiologists are trying with their best to combat with these pandemics. Why I have mentioned this? Because it is directly related to immunology and today's subject. So that is what the importance of immunology is. And at the outset only, I would like to mention some of the aspects of immunology that generally we ignore. As I am saying in the beginning only, that when we talk about antigen-antibody reaction, it is not only recognizing a specific antibody, but we can use it for several purposes, like right from diagnostic, then knowing the allergic reactions, then treatment, then prophylactic, and so on. So with this short introduction of immunology, let's move to today's topic, that is a major histocompatibility complex. As you can see, I have divided this particular topic into this particular chapter, or this particular unit of that is allotted for the SPPU, that is Savitribai Phule Pune University. To this particular unit, I have divided it into 2 lectures. So first 2 topics: structure of MHC in man and mouse, and structure and function of MHC class 1 and class 2 molecules, that today in the first lecture I'm going to deal with. And in the later session, or day after or sometime, lecture 2, that is that I will be dealing with the polymorphism of MHC. That is again very interesting to understand, and MHC antigen typing. So with that, I will start with structure of MHC in man and mouse. So of course, with a very short introduction, let's see what this major histocompatibility complex, that is widely abbreviated as MHC, is, and why it is called as MHC, and what are its implications, and how important it is in immunology. So of course, we all know the primary function of our immune system is to recognize, to find out the foreign substances that we call it as antigens. But let's talk in general way, that the very obvious function of immune system is to recognize and remove, eliminate, degrade foreign cells and antigens from the body. To do so, there are several specialized, expertise subcell population in our body. So of course we can't help it using abbreviations and short forms in immunology. So I take this I take it granted that basics the students might have or must have studied as you are in the class of 3rd year BSc. So T cell, B cell, and their subpopulation, and their types, and their functions, and their expertise, and their specialties, such basic things I may take granted sometime. So T cells and B cells are they are involved in immune system, as we know. So they work on the basis of that they recognize surface molecules which are foreign, and we call them as antigens. It is all right with the B cells that proliferate to give plasma cell and then produce antibodies, because antibodies can recognize intact proteins. Of course, intact proteins means the degraded product of proteins, that means the peptides. Again, it is granted that when I say protein, then it's degraded protein. So T cells and B cells, they recognize, of course, the foreign substance when it invade the body. But this foreign substance, if they are masked, if they are inside the cell, there may be a problem. It is all right with antibodies that recognize the intact peptides, by binding the epitopes. That epitopes, that means the actual part of the amino acid or the proteins, that may be linear or discontinuous, and brought together to be identified, to be recognized, and to be acted upon. So it is all right with antibodies to recognize with. But when we talk about T cell receptors, how T cell recognize these body substances? Of course, T cell do. So T cell for recognition, T cell do not work like that, as I mentioned in antibodies. T cell receptors recognize only linear epitopes generated by degradation of the intact protein that is either engulfed or that is inside the cell. That is we call as that foreign substance, foreign body, foreign antigen, foreign cell, whatever, inside created and what is called as antigen. So it is to be processed because T cell receptor recognize only linear epitope. So intracellular pathogens like viruses, tumor antigens that remain hidden, that remain masked within the cell and hence not available for recognition. That creates a problem. That really poses practical difficulty. So this practical difficulty, because in the earlier only we have seen that T cells and B cells use surface molecule. They have to be surfaced, whatever to be recognized. So if they are internalized, that poses a problem, and this problem is solved by displaying these internalized or internal degraded foreign substances or foreign, of course, proteins, pathogenic parts, antigens. So they are displaced somehow on the surface of the cell. They are present. How they are displayed then? These antigens, these degraded products, or these antigen pieces are held tightly with some binding moiety, a specific structure where it very specifically binds with that particular antigen. We call it as a groove of the cell surface protein, called cell surface protein of kind. That cell surface protein is called as major histocompatibility complex, MHC. We abbreviate. This is a standard or accepted, universally accepted abbreviation for that. So this protein, which manages to display these antigens on the surface, are called as major histocompatibility complexes or major histocompatibility complex molecules. And as these are proteins, I said, these are again coded by some genes. So they are encoded by a cluster of genes, rather many genes, more than 100 of the genes totally. The code they are encoded by. These genes are called as MHC locus. We will come to read later why MHC and what are their other names and how they differ in animals and all. So till now, I hope it is clear to you that what is meant by MHC and how they are coded by a number of genes. So these molecules that have solved the problem we have mentioned in the discussion, the problem posed by that internalized molecules that has to be, if they are to be recognized, they have to be displayed on the surface. So the task of displaying on the surface is taken up by special kinds of proteins. We call them as major histocompatibility complex. So these major histocompatibility complex proteins are generated inside the cells. When they are generated when such antigens enter and digested. We in general term, but we can say they are processed. And the complex of this digested or degraded foreign substance, antigen plus MHC, this particular complex, and as it must be noted that the binding is again specific. So these MHC molecules and antigen which is processed inside the cell, these MHC molecules which are produced, which are generated inside the cells upon the entry of that digestion or that particular antigen, the sequence is like that particular antigen get internalized, or the cell or virus that get internalized, then they are degraded, and then these MHC molecules are generated. These MHC molecules specifically bind with the antigen, and they display it on the surface so that these antigens could be recognized by earlier mentioned T and B cells. Okay, so this is how the problem is solved. So MHC molecules that act as cell surface vessels actually. Okay, for holding up these molecules to be displayed in the form of fragments or digested or degraded products of isolations, and so that they could be recognized by T cell receptors. Sir, I'm audible?
Yes, sir. Yes, sir.
Intermittently,
You're
I may
audible,
keep
sir.
on asking.
Go ahead, sir.
Okay.
Yeah.
Yeah. So, question arises: why this MHC got its name? Why MHC? I mean, the name is given to these genes. As we said, we have defined that MHC locus code for a protein called as MHC protein. Now, why this is called as MHC? Because this code for proteins responsible for rejection or acceptance of a particular transplanted tissue. Now, question arises: why? I mean, what is the relation between this? What is mentioned here is tissue and graft and transplantation. So, since the term histocompatibility, so histo means pertaining to the tissue and all, we know, and compatibility, whether it is to be accepted or rejected. So this particular loci, they are responsible for such a protein which may determine whether the transplanted, whether the foreign substance, whether the foreign tissue will be accepted or rejected. So they are involved in that, and hence the name MHC given to it. So tissues or organs or another. Sir, I'm some messages popping up. What is it? Should I view it and admit what? 3 people.
Yes, sir. At the end
I
of
just
session,
ignore?
we will take. Yes, sir. At the
I
end, we
should-
will take the question answer.
Okay.
Yes,
okay.
sir. You should continue, sir.
Okay.
Yes.
Okay. So tissue are organs of another individual of the same species or of the same individual are recognized as non-self. Of course, they have to be. So, and hence rejected. So how it is being decided whether to be accepted or rejected, and this is decided, this is determined with the help of these molecules. Hence compatibility, histo, because tissue related, the name may be. A very interesting work was done by Peter Gorer in 1940s around. He was studying some antigens responsible for this kind of graft reactions, and he also studied the blood group system in mice. While studying this particular graft related phenomenon and blood groups in mice, he came up with a very interesting observation. We have divided into 2 parts. So in the beginning, in the next slide, we will see that particular picture, how he came up with the conclusion. But what Gorer worked for this, or with concern to this particular phenomenon, is that he found 2 blood groups or antigens rather, let's not say blood group, 2 antigens in the mice. Let's say A1 and 2. So antigen 1 and antigen 2. And since he was studying the grafting tissue and several things, he observed that one antigen is all right. It is common to almost all of them. But the other antigen was actually responsible for rejection of the this particular reaction or this particular phenomenon they are studying. And he called it as, since it is concerned with that of histocompatibility, he called it as H1 and H2. And first antigen that is common, so the second one, that is H2, is associated with this rejection, and hence it is called as H2. And hence in mice, it is referred to as H2. So the histocompatibility antigens are cell surface antigens in that case, that induce an immune responses leading to the graft reaction, graft rejection. So H2 antigen system was found in major as a major histocompatibility antigen for mice. Okay, and this is what he found was a multi-allelic cluster of genes, and then referred to it as major histocompatibility complex. He also now we know that various loci of this complex was elucidated, and then work of Jean Dossey and then Snell developed a very particular, define a very nicely the phenomenon. So they were also awarded Nobel Prize in the 1980s for their work. So let's move. I may ask Arvind sir.
Yes, sir.
So this is what a short story of antigen 2, and this one was how it was called as H2 antigen and major histocompatibility complex. And I got stuck with this particular. I didn't understand why. Arvind sir, I got stuck with presentation. I'll try to resolve the problem. I cannot move ahead. Arvind sir.
Yes, sir. Yes, sir.
Just
resolve it, sir. Yeah. Are you using a mouse, sir, or you're using a keypad of your laptop?
I'm not using mouse, sir, but now it's working.
Okay.
So we were here. Sharing. Is it visible now?
Yes, sir. You're sharing. Yeah.
Yeah. So, dear students, we were with this particular workout of why it is called as H2 system in mice. Okay, the work of Peter Gauger, and Snell and Jean Dossey. Now, what he was studying, he found that there is a group of 4 genes, 1 to 4, and he identified while working on these tissue reactions and blood grouping. And further, he conducted, along with Snell, Gauger conducted a very simple experiment. In that, he inbreeded 2 mice. As you can see here, for the simplicity, I label it as D and K. So H2K and H2D, we all know. So for what purpose he wanted to study how and why the rejection takes place if the skin is grafted. So what he did, very simple experiment. With this simple experiment, he showed that antigens coded by group 2 genes take part in actual take part in this rejection of T1 or skin transplant. And what he did, as you can see here, in F1 generation, since the haplotype is of D as well as K. For simplicity of microbiology students, I may define haplotype is a set of allele that come from one of the parent. So this F1 generation contain both the haplotypes. So we concentrate here. If this F1 generation mice allowed to undergo a transplant, say skin graft or something from any of the parent, the graft is accepted. Very simple reason why it is not rejected, because this F1 generation contains both the haplotypes, and any of the parents' graft is, if planted here, that haplotype is already present with this F1 generation mice. But reverse is not the correct. If the skin is grafted from F1 generation to any of the parent, it is rejected. You can see here in this, just because one of the haplotype is absent in any of these two parents, and hence it is concluded that that particular gene is, if it is present or absent, it matters in between two transplanting individuals, that is donor and recipient. So this major histocompatibility complex in human, it is called as HLA. Question arises now, why HLA? I will be coming to it. In mouse, it is called as H2, and rest of the animals we don't have to see what is it called as, but just I just mentioned. We have seen why it is called H2 in mouse, but let's see why it is called HLA in mice. But before that, students, you always keep in mind that MHC locus, this gene, is found on short arm of the chromosome number 6 in mouse. Okay, so chromosome number 6 of human and chromosome number 17 in mouse. Sorry, so it is present on in case of humans, it is present on the short arm of the chromosome number 6, and in mice it is in 17. I'm insisting this because I wanted to again mention while studying MHC class 1 proteins, I'll come back to it, how some of the part is also distantly present. Anyway, that I won't mention here. So why it is called as HLA in humans, it is given here. Because previously it was observed that this particular gene is, or particular antigen is present on white cells. So we have seen why it is called H2 in mice, because H1 and H2, these two antigens. Okay, but in humans, the gene product earlier found to be associated with white cells. Of course, later we come to know that all the nucleated cells do contain this particular antigen, and hence, as it was found earlier that in case of humans, the antigen was present on white cells only, it was called as human leukocyte antigen, and the name stuck to it as HLA. Even today we refer to it as HLA, but actually you should mention it as MHC. So that is why the names are given. I hope you have got the concept why MHC, why HLA, why H2. Now let's move ahead. So MHC molecules are coded by individual genes and not it is not the product of the rearrangement of genes as that happens with TCR and BCR. B cell receptors and T cell receptors. Neither do they undergo any somatic mutations. So like the other cells, germline rearrangement, it's not like that. So MHC gene do have that particular property. So the diversity still exists. Now this diversity is because of enormous number of very huge number of combinations that are available among these more than hundreds of the alleles, genes that are present. And that is why we will be looking into the thing that every individual is different from one, even in the same species. This is just because of this diversity. There is no identical set of these antigens into individuals. I said set, there may be antigens, but combination, the set of antigens is always different. And that is why antigenically we are different from each other. How much foreignness is there, that will be determined whether the graft is rejected or accepted, whatever the type of graft may be. So diversity. The MHC genes are recognized three genes rather. They are organized by on these three genes. They are referred to as MHC class 1 gene, class 2 gene, and class 3 gene. I'm sorry here not to mention as class 2. It appears again class 2. Class instead of class 3, it appears class 2. So MHC class 1 gene is present on all nucleated cells, and it presents antigen to intracellular microbes. It presents antigen of intracellular microbes to TC cell with CD8 moiety, and that mediates of course TC, cytotoxic T cell, and that mediates CMI. This is a very simple formula for that. So I'll repeat, MHC genes are organized in three different regions. For the sake of understanding, we have seen these three regions. Which three regions? MHC class 1, MHC class 2, and MHC class 3. Now, MHC class 1 genes code for a protein. We call it as MHC class 1 proteins. Okay. And what are these? What is the function of these genes? And where are they present? They are present in almost all of the nucleated cells, and mostly on the lymphocytes. And what they do recognize, or they do present this, as we have already seen in the introduction only. What is the function of MHC? The function of MHC is actually rather than saying function, they have solved the problem of internal antigens. To be recognized, this antigen must be displayed at the surface. So the basic function of MHC is MHC gene product is not only the compatibility and rather histocompatibility and graft rejection and determination whether the graft will be identified or rejected. This is not the original function. Rather than MHC molecules are associated with this presentation of the antigen to the T cells. Okay, so MHC class 1 antigens, so they do accordingly. What they do, they are present in all of the nucleated cells, and they present this antigen, exogenous antigen, to the CD8 type of the cell and stimulate the CMI. On the contrary, MHC class 2 gene, they are only present in the very specialized, expertise kind of defense cells like macrophages, APCs, CDs. Okay, so they are only present on the antigen presenting cells. And MHC class 2 gene product, what they do, they present the exogenous microbial antigens. Exogenous, though I'm saying exogenous, we have already seen that this exogenous bacterial or whatever invading pathogen, it is taken up by it is taken up inside the cell. So the problem is that this taken up moieties has to be expressed to be recognized. So that displaying is done by MHC class 2 gene product. So they present these exogenous microbial antigens to TH kind, CD4 moiety, and they mediate human antibody response. So examination point of view, if you keep this equation in your mind fixed, it will be easy to mention in your answers, and evaluator will easily be understand that okay, the student have got the concept of MHC1, what it does, and what is MHC2, and what it does, where it does, and how it does. So this is, I have simplified the formula, what MSc class 1 might do, what MSc class 2 might do. Now, MSc class 3 has nothing to do with this displaying and it, but MSc class 3 gene product is very important function. There are actually many functions carried out by this MSc class 3 moiety. Just for the sake of information, like it may be involved in several like complement you might have heard about. Okay, so complement system and several security products of the immune system, cytokines, and so on. Okay, so MSc class 1, class 2, and class 3 genes, their function, how they function. Okay, should we move ahead, sir? So answer.
Yes, sir.
Should we move ahead? I just intermittently I keep on asking to somebody. Yes, sir. So students, I hope that you have got the importance of MHC and MHC class 1, class 2 molecules, and how they are important in the seminal role they are playing in displaying the antigens of both the kinds, for the exogenous antigens and endogenous antigens. MSc class 1 deals with endogenous antigen. MSc class 2 gene product deals with exogenous antigens or microbes. I particularly take one more minute here to mention that this is not the these are some of the selected genes or their alleles we are studying about. For example, MSc class 1 gene product may also be involved in some other extra functions that we refer to it as non-classical alleles or genes. I'll come to it later. So I will move ahead. We must know some more things about MHC. So this must be clear to you that these are the molecules, proteinase in nature, which are the product of MHC genes located on MHC locus, which is tightly lined cluster of genes in all mammals. Even it is present in certain invertebrates. Rather, it is being said that origin of MHC gene might have took place in some invertebrates, starfish, and IR genes actually evolved from starfish. It has been said that. So invertebrates may have the correlation with like that. We may go as back to the invertebrates state. So every mammal do have this MHC gene, and this may be repetition of class 1, class 2, and class 3 genes, but in different angle. There we have seen the equation, how it works, where it works, and what it works. Here, class 1 gene contains what that we have mentioned. So class 1 gene, MHC class 1 gene, and their products are HLA, B, and C. While class 2, HLA, DP, DQ, and DR. And MHC class 3 gene codes for several products which are important in MAC, that is membrane attack complex or complement cascades and all. So let us concentrate on MSc class 1 and class 2. So MSc class 1 gene have these 3 particular loci, A, B, and C. I was mentioning something about additional aspect. So MSc class 1 and class 2 genes are not only responsible for displaying the antigens at the surface so that they can be recognized by the highly specialized immune cells. But here, for example, In addition to this ABC class, there is one other ABC genes. There is one gene. There are many, but I would just like to mention one. Say HLA G gene, which is actually responsible for protection of the fetus from the immunological system or immune system of the mother. Because don't forget, for a mother, a fetus is non-cell. So G gene is very meticulously working for that, and that is not a topic of our discussion. But these genes are also having some other functions as well. Class 3 genes are associated with some other functions like complement and all. So it must be clear to you that T cell can recognize antigens epitope. The immunologically active cells can recognize these antigens only when they are presented in the context of self molecules and at the products of MHC genes. So it must be clear to you that the highly active immunological cells, T cells, can recognize these antigens only when they are presented through these MHC genes in the context of cell molecules. Why I am emphasizing here, this is the another important aspect to be noticed, that self and non-self discrimination is also made by these MHC molecules. So it is not a good thing to have raised immune response against our own cells. So self and non-self or missed self. In some books it is the term given that self antigens and non-self antigens. So immunological reaction is expected to be raised against only all non-self antigens. So this discrimination is done that the T cell will recognize if and only if the antigen is presented by their own cells carrying their own cells MHCs. So and they present it with the context. Hence, the main function of MHC molecule is to bind very huge number of antigenic epitopes around the cell, in the vicinity of the cell, it may come across of pathogens. So, and present them to the T cells, which then get activated and induce either CMI or humoral immunity. Okay, either antibody response or cell toxicity. So MHC genes code for cell surface molecules. To summarize these 3 slides. So MHC genes codes for cell surface molecules called HLA. We have also seen wide HLA, which are so polymorphic. Their diversity we have seen. They are so polymorphic that virtually there are no 2 human beings having the same set. I'm saying set of HLA antigens. From this sentence, you may imagine how diverse, how polymorphic, how pleomorphic the MHC gene combination could be. Now the main aspect of MHC molecules, their molecular structure and function. MHC molecules are studied. They are separated, they are isolated, and studied with the help of the very sophisticated techniques like X-ray crystallography. And so their 3D structure is known, and we will see its ribbon structure as well. So both the classes, class 1 and class 2 molecules are having similar structure. Both of them are glycoproteins. Both of them are heterodimers in nature. This is the common feature of them. And expressed on membrane. You must, dear students, you must take it to the note that MHC molecules do appear at the cell membrane. They do present the antigen to the surface. That means they must be protruding. Some part of them are protruding along with the peptides they are carrying, and they wanted to show to T cell. So the protruding part that comes out of the cell along with the antigen, so that T cells can recognize these antigens. And some part of this MHC, it remain bound to the membrane of the cell. They are present. we all know that MHC are present in almost all of the nucleated cells. So this is the interesting thing to know about. This become more interesting when I will show you the diagram. But since we are discussing only the basic or the introduction of structure and function. So the part that comes out, the protruding part is called as N terminus, while the other end is called as C terminus. N terminus is as its name, C terminus is a constant. N terminus is variable amino acid sequence, and depending upon their sequence of the amino acid and their epitopes and their structure and their number, they can bind that specific antigen. So this structure that is protruding out of the cell membrane have a specific kind of groove, or we call it as a cleft, a groove structure where this antigen is present and displayed for recognition. So the peptide get associated with MHC molecule and are presented to the T kinds of cells, TH or TC. And the sequences near the cell membrane, that means the one that is not protruding out, here you can see. This is the N end, this is the C end I was talking about from the membrane. It is protruding, this is this protruding one that comes out. Actually, protruding means some part remain associated with the membrane and some very little part is inside the membrane. So this particular groove is associated with binding of the antigen. So here is the antigen, this is the MHC, and this is the cell, nucleated cell. This is how the antigen inside the cell was displayed to here, displayed here for the recognition by T cells. I hope you are getting it. Coming back to the slide, since we are trying to understand the function. So the peptide get associated with the MHC molecule and they are how displayed and presented. So the sequence near the cell membrane have here, that means near the membrane. These all are, this is a protein molecule. So it is made up of amino acid. I'll come to it, which amino acid, how many amino acids, what's its molecular structure. That is what we are going to see. But the amino acid sequence here is conserved. It do not vary a lot. But here it varies, variable region. While discussing this or while I'm mentioning this, you might have imagined what kind of molecule it is. It is similar, students, it is like a immunoglobulin molecule. Okay, the antigen binding region, and this is the FC and FAB. Okay, so the CD4 molecule get so accordingly MHC1 molecule presented to the CD8 and MHC2 molecule present to the CD4 subset population of the T cell. And this is the general structure. This is not this you can use this particular figure for your examination point of view. But the next paper I'm going to show you will be better if you prepare with that figure. This figure will give a very general idea about what the MHC molecule is. Okay, it consists of alpha and beta. That will come into it later. This is what about the structure, what is mentioned. So MHC1 molecules are expressed, as I said, on almost all nucleated cells. It is not the case with MHC2 molecules. MHC2 molecules are expressed on only the expert specialized kind of cells like APCs and Dists, dendritic cells. So almost all lymphocytes, they do consist of this. Both of them are heterodimeric glycoproteins. Both of them are proteins. Okay, but let us see as the title of the slide is MHC1 molecule, class 1 MHC molecule. So let's talk only about class 1 molecule. So it is a heterodimer again. It consists of two dimers, alpha and beta. Alpha chain consists of three subunits: alpha 1, alpha 2, and alpha 3. It is, and beta 2 chain, microglobulin chain. So alpha and beta, there are three parts for alpha: alpha 1, alpha 2, and alpha 3. And alpha 1 and alpha 2. Alpha 1 and alpha 2. This forms here alpha 1 and alpha 2 domain are actually these are the sites where the peptide, that is antigen, binds. While alpha 3 and beta 2D, while alpha 3, you can see here, and beta 2D there, beta 2 micron mu is as I have mentioned, is a kind having a kind of amino acid sequence which is conserved. Okay, so it is like a FC region of the immunoglobulin. So alpha 3 region is anchored on the membrane and we do not take part in antigen binding. So both alpha 3 and beta 2 support the structure rather than recognition. And each of these regions, the size is about 90 amino acids, and beta 2 is encoded by highly conserved gene. See here, it is present on chromosome number 15. So beta 2 is, we are talking about MHC1 only. Beta 2 is required for obtaining a fully folded alpha 1, alpha 2 structure and structural conformation and structural stability. So beta 2 has no role in recognition and presentation and specific binding and all. Okay, and beta 2 and alpha 3, hence they are homologous in structure and in function as well. So MHC1 molecules molecule cannot leave endoplasmic reticulum unless it's bound to the peptide. Once it gets bound to the peptide, they then only go and protein. I said this is the structure. If the beginner wants to know what is MSC, fine, you can draw this structure. But this is called as ribbon structure of MSC class 4 molecule. Again, this is not a good structure to be remembered for your exams. The next figure I'm going to show you, you can use this for your presentations in exams. If you present it like this, the evaluator will easily understand that the student have gone through and he is very much clear about the things. About which things, see here, we can say that dimeric structure, which dimer, the alpha 1, the alpha chain, the beta 2 mu chain, okay, 90 amino acids each, alpha 1, alpha 2, alpha 3, okay, 30 amino acids built up of this alpha chain, we can say, part, which is actually inside the cell membrane. This is the cell membrane. And the part which is embedded in the membrane is of around 25 amino acids. So 30 amino acids, 25 amino acids, and 90 amino acids. Collectively 45 kilodalton weight. On the contrary, beta 2 chain is only of 12 kilodalton weight and corresponds to alpha 3 amino acids. So this is what the structure of MHC1. I hope you are getting it. Chavan sir, should we move ahead?
Yes, sir. Yes, sir. Go ahead, sir.
Yes, sir. Yes, sir. Now let's see what MHC2 molecule is. As I said, both of the molecules are proteinic in nature. Both are dimeric. See, this is what MHC2. I will show you what we have seen in MHC1. This is MHC1 molecule. Okay, where you can see here beta 2 mu. This is a mirror image actually of this. Here beta 2 appears here somewhere. But MHC2 molecule is like this. There is no beta 2 mu. Alpha and beta are almost similar. They are bound with there is a covalent bonding. In here also there is a covalent bonding. Here also there is a covalent bonding. So there are so many differences. Eventually you may come across, but it is good for examination point of view if you keep on noting the things. What are the differences? Because if the question maybe like that, what is the state the difference between MHC1 and MHC2? Then this will be helpful to you. So the major difference is that it has a whole beta structure, alpha structure. Okay. So the chain has got two external regions. Again, MHC2 molecules are also dimeric. glycoproteins consisting of two different polypeptide chains, alpha and beta, associated with each other on non-covalent reactions. Non-covalent bonding is there. Each chain has external regions and embedded and cytoplasmic region, just as we have seen with MHC class 1. The two external regions are here alpha 2 and beta 1 and beta 2. So this is what the binding groove, alpha 1, alpha 2 and beta. Okay, alpha 1 and beta 1 actually is involved in binding, while this is constant conserved sequence, and this is inside C end and this is N end. So alpha 1 and alpha 2 and beta 1 and beta 2, these are the two regions. So alpha 2 and beta 2 are closer to membrane, and they are the structures that have nothing to do with binding, but they are conserved amino acid sequence and thus belong to the Ig superfamily, like immunoglobulin-like structures, as I mentioned earlier also. And here alpha 1 beta 1 form peptide binding cleft. As I mentioned, this is the part actually which are involved in specific binding of the antigen, so that it could be presented. So expression of class 2 molecule is restricted to APCs such as CDs and macrophages. Unlike that of MHC1 molecules, all nucleated cell contains MHC1, but it is not like that. It is associated with FACs, antigen presenting cells, and dendritic cells only. Okay, so since they are heteromeric and MHC2 genes are both the chromosome homologs are actively transcribing, so there may be different combinations. So alpha chain can get associated with beta chain from the same chromosome, and there are many combinations, and that is how diversity may be created. So MHC class 2 molecules present antigen to the EHL, and further reaction, as we know, it causes HI, humoral immunity. Hope you got it. C end, N end, conserved sequences, antigen, it's a bit of binding region. This is the detailed structure of beta 2. For examination, you can remember this structure. Easy to remember, easy to draw, and covers most of the important properties like what is the molecular weight of this region, how much the region is, where is the conserved sequence, what is the region that is inside the cell, what region comes out, where is it cleft. This is the site where antigen is bind and presented. Hope you are getting it. Okay. This structure I have purposely shown you to have a comparison of MHC class 1 molecule and MHC class 2 molecule. So both are having N and C end, conserved sequence, and this is the sequence of amino acids of the chain that is actually binding the antigen and presenting it. So you can have this for comparison. More elegant structure of MHC class 1 molecule and class 2 molecule. You will find it in Kuwait. But I have not shown it earlier because it's a little bit difficult to draw as far as examination presentation is concerned. But this is good to understand. So where is the groove? Here is the groove where antigen sits. Which is the conserved sequence? These are the conserved sequence. Where is the membrane? How much the molecule is within the membrane? This much is inside the membrane, and this comes out, protrudes out the distal end where antigen sits and presented to the T cell. So MHC, and this is the MHC molecule, the antigen of this particular cell where the MHC is produced of the cell. This particular structure will only be recognized by T cells. T cells will not recognize, will not bind particulate antigen somewhere, just randomly present. And this will bind here only. This binding is very specific. No own antigen can bind here, and that is why it can be discriminating between self and non-self. So summarize, both class 1 and class 2 molecules are present on nucleated cells. Class 1 antigens are present on all kinds of nucleated cells, repeatedly and so on. It is not the case with class 2. Okay, class 2 antigens are present only on immunocompetent cells, specialized cells, B cells or AP cells, and class E below. It is associated with the stuff, complement and all. These are the genes, MHC genes, as we have mentioned. Let's not go in much details about what is the case with mice, but let's see HLA. In humans, it is called as HLA, no? The name stuck up, as I said in the introduction only. Actually, we should call it as MHC, but the name HLA is stuck up because previously it was thought that since they are present on the leukocyte, and we are calling it as antigen actually, and they are not, the main function of them is not to be agent for graft, but rather their main function is to present antigens for the cells which are immunologically competent. So this is the gene structure. Class 1 gene, you can see A, B, and C. As I said, though it has been insisted in all the books, 3 genes, there are many genes, I said. For example, G gene, there are so many other genes, they have some other function. G gene, as I said, it is responsible for protection of the fetus from the mother's immune system. And there are actually many, but that is not the part of today's discussion, since we have to concentrate on MHC gene and there is compatibility structure. Class 2 genes, as I said, PQ, R, PQ, you can see here, DR, okay? And class 3 genes, which are responsible for MAC and complement cascade proteins. Again, this is simplified gene structure. This last slide will show you the overall summary of structure and function of MSC class 2 molecules. So this is the cell, nucleated cell. Okay. This is the MSC class 1 molecule. This is the groove where antigen sits. This binding is specific. This is beta 2 mu, which gives stability of this whole structure. So this antigen, which were rather present here, is displayed here so that could be recognized by T cell of CD8 type, and this CD8 molecule recognizes it. Then this T cell get activated and produce its effect, cytotoxicity. Similarly, to summarize, this is the cell, nucleated cell, okay, where this antigen would have been here somewhere, now processed, presented here by binding to the MSC class 2 gene, alpha chain, beta chain, and then recognized, since it is bound with MHC molecule of the same cells, these two are of same host cells. So CD4 binding confirms, and this T helper cell, Th kind of cell get activated and stimulates immune response, that is production of antibodies against this antigen. So that is all about how MHC have solved the problem of recognition of this red color antigen, which would have been otherwise inside the cell and would be difficult to recognize by these immunologically active cells. So these are my references, some of the reference papers and some books. Thanks a lot. Any query or some discussions? This is just for part 1. In part 2 lecture, we will be studying other aspects of MHC class 1, class 2, and their typing and all. Thank you very much, students.
Thank you very much, sir. Thank you for a nice presentation on MSC, both the molecules, MSC class 1 and class 2. You have elaborated it very lucidly. I hope this topic is definitely benefited to our UG, PG, and those students who are preparing for the net sets. Now,
Yes,
this topic is open for discussion for the participant. I thought that few of the participants have personally, Wale sir,
yes,
some of the participants have personally texted you some question or queries on this topic. I hope firstly you have to go through that queries, and later on we will see if
Yes.
those participants having live having the queries, they may ask live in this session.
Yes, sir.
Yeah.
Okay. Okay. So if they can, if they have some question to ask directly, they can ask as well.
Yes, sir. Yes, sir. Participants are requested to ask their queries to Dr. Wale, sir. I thought, Mr. Vivek, do we have queries?
I can read out. I will answer these queries. Those have sent me on questions. There are several questions I could see,
Yes.
sir. But if they don't have question, I think we will stop. Thank you so much, sir.
Thank you, sir.
Okay.
Now, yeah. Now I call upon Mr. Venkatesh Jadhav, sir, my colleague, to propose the vote of thanks for the second session of the first day for this countrywide classroom program. Mr. Venkatesh Jadhav, sir, please.
Okay. Thank you, sir. Thank you, organizer. And I would like to thank Dr. Rajan Wale, sir. So, sir, the talk you have given was really knowledgeable and thought-provoking in case of their diversity, their MSc molecule structure, and how it is presenting any antigen to activate our immune system. So now this topic, I would like to say this is a very you have touched and enlightened a topic which is a very relevant to present COVID-19 situation. So it is a very impactful topic to understand how immune system is get activated in response to a different antigens. So as we know, the COVID-19 is also is treated as an antigen to get synthesis of antibodies, which is not possible without proper functioning of this MSc molecule. And one more point I would like to highlight from Sir's topic, that your language and your presentation was so simple to be understand by student, and it which is attracting a participant to do not detach. So I would like to again thank you, sir, for your cool, calm, and very informative, current situation oriented talk on MSc molecule in immunological sciences, or this branch. So I hope our students and all participants from different areas, different courses will definitely benefit from this talk. So I would like to again thanks, and I would like to again thanks organizer for giving me a chance to enlighten the sir's talk. So thank you, sir, and thank you, organizer.
Thank you very much, sir.
Thank you, Venkatesh Jadhav, sir. And once again, I thank you. Thanks, Dr. Rajan Wale, sir, for his talk over the MSc complex molecules. And hope to see you again in this countrywide classroom in the next week, as per your schedule.
Sure.
Yeah, friend. Yeah, for
Yes,
today's,
sir.
I declare that this session is over. Tomorrow's session will be started at first session will be at the morning 10 o'clock, and second session will be at the noon 2 o'clock. So I request all the participants to tune with on the scheduled time. Thank you very much. Wale sir, I will call you personally.
Yeah, sure,
Yeah.
sir.
Yeah. Yes.